So for the next about 14 minutes, where's my, sorry. So I changed my title a little bit because I wasn't aware I was given a title on basic research in vascular remodeling. So I will tell you very briefly about our recent publication in circulation research about this one important peptide, the proteins called UCP2 on the mitochondria, serving as a mechanosensitive inhibitor of atherosclerosis. Now, we know the blood flow along this vascular tree, and particularly in this straight part of blood vessel, we call this laminar flow, which actually is vasoprotective. When the flow meet the somewhere, we call this a branching point or curvature, and this flow pattern turn to be turbulent, and those are not good, causing harmful impact on the vascular function. And very briefly, now these two patterns of blood flow has a very distinct impact on the phenotype or this feature, or the typical feature of endothelial function. And for example, this laminar flow, normally vasoprotective because the basal level of oxidative stress, the expression of inflammatory, the molecules are normally low, so that's why very tight between those endothelial cells and preventing this leakage. And those are good, something we want to preserve. On the other hand, and the flow at this curved blood vessel, there's a branching point. Those endothelial cells now express high level of inflammatory biomarkers and the oxidative stress, of course, and also this orientation of endothelial cell disturbed and very different. So that might explain why this and the plaque formation may occur now in this curvature or this branching, this bifurcation of blood vessels, because those areas express high level of inflammatory biomarker, which is the most important early the triggers now for the development of atherosclerosis. Now, for long years, people know this phenomenon, of course, and then a lot study. And if someone interested in this mechanism stimulation and the related to changes in vascular function, and this has been known for quite a long time, and that we started pay attention to this actually is our first publication. We got lucky in late the 2016. But today I do not have much time to tell you much about this. But in recent years, we spent quite a lot of time trying to dig up more information about how endothelial cells respond to different types of shear stress. But today I want to tell you one story about this uncoupling protein 2 on mitochondria. Now, actually, about 10 years ago, we published a paper in Circulation Research. At that time, we just happened to get to realize that this UCP2 can be very important because this control mitochondrial reactive oxygen species production. And we use those DBDB, the diabetic mice, and we found if you overexpress UCP2 and very effectively preserve endothelial function. And this is quite interesting. Later on, we found this able to lead to the reduction in the ER stress, for example, and lower the oxidative stress and increase nitric oxide bioavailability. But no one ever known whether or not this UCP2 can respond to different stimulation in terms of this shear stress. That's actually we started a couple of years ago by a PhD student, Regeneration, now is working in Shanghai Jiaotong University. Now we have this setup called this flow chamber. That means we can mimic different pattern of flow. Now just give you an example here. We have this oscillatory, this disturbed flow we call OSS, or this is unidirectional flow we call laminar flow. Laminar flow is good, and oscillatory or disturbed flow is bad. And here you can see very clearly. Now it doesn't matter you use a human this aortic endothelial cell or this venous endothelial cell, and the response is very similar. Now, if you subject endothelial cell to this disturbed flow, and then you're always able to see reduction of now this UCP2. Yeah. On the contrast, if you give a flow, the laminar flow stimulating endothelial cells, and then you will see the significant increase now in the expression of UCP2. So this is a very early observation, and then we realize this is something quite unique. But of course, because this is UCP2 is not a transcriptional factor, it must be something else, and that control the expression now in response to mechanism stimulation. So, but not only in the cells, we are able to demonstrate now this mechanism sensitivity of UCP2 expression, and also in the we dissect intact of aorta, and as I mentioned, this is aortic arch, this is thoracic aorta normally is straight, and this flow we call laminar flow, and this area, curvature, this is distal flow. But here you can see very clearly. Now in this is a straight part. Now we have a higher expression of UCP2. Now as the lower, in the aortic arch, you have a lower expression of UCP2, and that corresponds to a little bit increase in the adhesion molecule. So we can 1, for example, yeah. And of course, we need to identify how this UCP2 expression is controlled. And then we screen quite a number of transcriptional factors which known to be mechanism sensitive. And then we narrow down to this KLF2. And actually, this is a very important one of master transcriptional factor expressed by endothelial cell, controlling particularly the eNOS activity. Now, releasing nitric oxide, those are extremely important. The gas molecule released by endothelial cell controlling now this local blood flow. But here you can see very clearly, now we are able to overexpress, for example, this KF2, and then you will see now KF2 can reverse now this disturbed flow induced reduction of UCP2 expression. On the other hand, if you silence this KF2, and then you can normalize or reverse now this laminar flow induced UCP2 upregulation. So these are very clear. Now this KF2 actually is one of one of those upstream the regulator of UCP2 on mitochondria. Now also we spend quite a lot of time trying to understand how these things are regulated. If you are interested, you can read our recent paper. And a few things, of course, I need to emphasize, as I mentioned, KF2 is a very important regulator of eNOS. And then we take eNOS as a control, and then you can see very clearly eNOS expression increase once you overexpress KF2 on endothelial cells, and together with UCP2. And that some data actually also support our earlier observation. At that time, we did not go deep enough. Even we published in circulation research, apparently UCP2 is quite important in regulating now vascular endothelial function. Now, on the other hand, of course, now you need to have a gain and loss function of those experiments whenever you submit a paper, particularly if you submit a paper to circulation research, that's very tough. And then this is normally we do. And then one hand, if you here, you can note down the KF2. It doesn't matter by which way, you're always able to see now reduce expression of KF2 not only, and also UCP2 together with the eNOS. Yeah, the genes, the name called the NOX3 actually is encoding the eNOS. Together with the protein here, you can see very clearly. Now, of course, why we want to study this, because we really want to identify whether or not we're able to identify any existing drugs. Yeah. And that we've been studying for a number of years, and we have been able to screen a few drugs, and I just give you 2 examples. One is a statin, which is a very the magical compounds, and I like very much. And another is resveratrol, that I like by everyone else. Yeah, if you like to drink the red wine, I don't know that's only derived from red wine, but certainly these drugs has a huge range of this pharmacological activity, and I think it's would be very useful tool for any of those studies. But here you can see very clearly now you can use the interleukin-1 beta, which is, can suppress expression UCB2. Now, in co-treatment, you can see very clearly able to reverse. Now by the treatment with rosuvastatin, actually we test a few those statins and also resveratrol within a reasonable concentration. Now, all this effect is very clear. So that means UCB2 can be regulated. Actually, our first paper was published quite a number of years ago. We found this is a DPP4 inhibitor, which increase now the GLP-1 that's used for the diabetes patients. And actually, it's in our SHR, the spontaneously hypertensive rates able to increase now UCB2 expression. Then we did not go further, but that was the first demonstration by any of those drugs now, which can raise UCB2 level. So this is, we feel quite happy because, study has been used to not only lower patients' lipids, and it has been used to reduce the plaque formation, and at least these are probably additional, beneficial effect if someone utilizes statin to reduce the atherosclerosis. Now, of course, you need to confirm this in the animal. We use ApoE animals and then inject with this AAV carried the expression of PCSK9. So rapidly, this animal have a higher this cholesterol level in the plasma without changing actually it's glucose, and they also rapidly enhanced formation of plaques. So here you can see very clearly now. Now IVO note down specifically UCB2 on the endothelial cell, and then you will see there's a further increase in the plaque formation. So that means somehow in our animal models show now this UCB2 might somehow actually contribute, at least regulate those physiological processes during the atherogenesis. But as I mentioned in our cell studies, because two different patterns of flow has a very opposite effect on the expression of UCB2. So we use this partial carotid artery ligation and then to generate this flow enhanced atherosclerosis. So here I do not need to go details, but here you can see very clearly. Now, if you do this in both wild type and another is with endothelial cell, now UCB2, the suppress. And then you can see there's a rapid increase or significant increase this formation of plaques. But on the other hand, if you use this Heron-5 promoter, and then you can overexpress UCB2, and in this case, use a similar model, and then you can see very clearly. Now this is the endothelial UCB2 overexpression can suppress now this flow associated the plaque formation. So for the good sake of time, and just a small summary for you. Actually, we spent quite a lot of time. We found this CAV2 actually able to directly bind to promoter region of UCB2 with certain lengths. And there's not direct effect. Actually, there's more effect involved, and then UCB2 able to link to APK, another very important kinase. And then also we found that FOX1, we have treating those animals with the FOX1 inhibitor, and actually achieving very similar effect as you overexpress now UCB2 on the endothelial cells. Now with this, I think this is an interesting study, and then because we identify some transcriptional factor which happen to be mechanosensitive, and that control this one of very important protein expressed by mitochondria. So now we of course wish to, if I have more people and then more space, I might carry on some other studies along this research direction. So thank you so much. So this is my new university, very small. If you got a chance to come to Hong Kong, please have a visit. Yeah, it's a nice university, but a lot of people actually very small. There's one big building, and Professor Wang Yibing actually is will come to visit my department as the academic advisor, and I will be very honored if Yibing will come in the early December, and then I will show him around, and then apparently, if we have time, I will give a talk to us as well. Thank you so much.